MedCram.com.
Well, welcome to another MedCram COVID-19 update. Today we're going to talk about why you might not have gotten COVID-19 as yet, and why maybe you haven't even tested positive. So I collaborated with Emily Lawrence on this piece, which I'll give a link in the description below to, and it's a Parade Magazine article titled, "If You Haven't Gotten COVID Yet, This Might Be Why." And we're going to review today the science behind this. There's one paper in particular that has not been published or peer-reviewed, but has some very interesting findings, and we'll go through that paper. And spoiler alert, it has to do with genetics and your immune system and a little bit of technicalities, so you have to be educated first before we can go through that. But speaking of education, who are we? We are MedCram. I'm Dr. Schwelt. I'm a board-certified internist, pulmonologist, critical care specialist, and sleep specialist with faculty positions at 2 Southern California medical schools and the co-founder of MedCram.com, where we record and publish continuing medical education videos on various topics in medicine, which are used by publishers, medical schools, and other healthcare professional schools as well. And my passion is basically explaining medicine clearly so you can make decisions. So let's get into exactly what it is that we need to know about and why some people, maybe even up to 1/10 of the population, are very well protected genetically from COVID-19. This is a potential theory here. So the first thing you've got to understand is something called HLA genes. There are 2 different types of proteins, actually more than that, but one that we're going to be talking about today called the MHC1 protein. That stands for Major Histocompatibility Complex Protein. And this protein is coded for by different genes, specifically the HLA-A, B, and C genes. These proteins are on the surface of every nucleated cell in your body. Minus the red blood cells, every nucleated cell has MHC1 proteins. You get 2 different copies. You get 1 copy from your father, and you get 1 copy from your mother. So you will have a diversity of different proteins. And your set of proteins in your body that are on the surface of your cells are very specific to you. And they're on every cell of your body, but they are very, very different compared to other people's cells. This is the reason why specifically you have to HLA match organ donation. Because if I were to take a random donor from somebody, let's say a liver or a kidney or a lung, and put it into your body, their MHC proteins would be very different on a random scale than yours, and you would reject those organs. So this is the reason why when they transplant organs, they have to make sure there is some degree of HLA matching. And there are tens of thousands of different combinations. So it's very different in everybody. But in addition to having these proteins which are so diverse, these proteins are essential in terms of signaling your immune system that those cells that have these proteins on them are infected. So let's read what it says here, and I'll post a link to this paper. The Major Histocompatibility Complex System, MHC1, 2, and 3, known as the human leukocyte antigen. The reason why they're known as human leukocyte antigens is because when we first discovered them, we discovered them on white cells, and we didn't realize that they were in other places as well. It's located on the short arm of chromosome 6 and contains the most polymorphic gene cluster in the entire human genome. What does that mean? That means that these things are so diverse between human beings, you've got to match them in order to do transplants. So they're very different. Furthermore, HLA consists of 3 regions, which have been designated as class 1, 2, and 3. We're not going to talk about 3 at all here, but we do show up here 1 and 2, and I've put that up there for clarity. Class 1 has 3 different gene category products. There's HLA-A, B, and C. And then for class 2, there's these DR, DP, and DQ. They go on here to say the main function of HLA class 1 gene products is to present endogenous peptides to corresponding CD8 positive T cells. What are CD8 T cells? These are the cells that go around the body and kill infected cells. So those are extremely important cells because if they can get their job done very quickly and very early, it can limit the degree of the infection. If these are very strong, the degree of infection in your body is very limited. If they are weak, this allows the viral infection to spread. So it's very important that your HLA class 1 gene products make a good protein in order to make sure that these CD8 T cells can do their job. Now, we won't talk about MHC2 protein, but if you want to be complete, these are the things that actually present antigens to the immune system so they can do their job. So they're important as well, but not for today's discussion. Today, we're talking about MHC1 proteins. These are the proteins that tell the immune system that a cell is infected. And just to nail home that point here, look at the number of HLA class 1 alleles. That means copies of those genes. 25,000 different ones. So you can imagine that if you wanted to do a bone marrow transplant, you would need to have a match right away so you could tell whether or not somebody could donate. And that's exactly what they do. When you donate blood to the blood bank, you're able to have your HLA genes put into a bone marrow registry so that if somebody ever needs bone marrow and your HLA code matches their HLA code, they will contact you to see if you're willing to donate your bone marrow to them, because that would be a perfect match. So you can see here the chances of that happening are pretty low because there's so many different types of alleles. You can see the class 2 and other non-HLA alleles. For HLA class 1, look at the number of alleles in group A, in group B, in group C, and these are other smaller gene categories, 7,000, 9,000, and 7,000. So let's show you what's actually going on here with HLA genes in the major histocompatibility 1 complex protein family. This is a cell of your body. It's not an immune cell. It's just a cell that has now been infected. Here comes the virus. It attaches to the ACE2 receptor, and it gets internalized. And as a result of that, it gets broken up by the cell, and it gets cut up into tiny fragments. And these fragments are proteins, tiny proteins, otherwise known as peptides. I'll show you this in a little bit more detail. So these tiny proteins get associated with your MHC class 1. That's here in red. And then they go to the surface and they present that to T cells that are going by. So it's almost like saying, hey guys, here's the signal. This cell has been infected with some virus that should not be here. And T cells, already pre-programmed to look for these different types of shapes that are kind of unique and not self, will come along. Here comes a cytotoxic CD8 T cell. So these T cells, which have CD8s on their surface, are well known to be cytotoxic. So basically, here comes a killer T cell and recognizes that this cell is not healthy and it's potentially a factory for more viruses and needs to be taken apart. And so what happens is it migrates, and this little green thing right here, which is the T cell receptor, binds the major histocompatibility complex 1, which is this red thing right here, and this little piece of the protein from the virus. This binding here can change somewhat dependent on the actual protein fragment. We know that. The part that's interesting to scientists now is this red part. This major histocompatibility complex protein, which is a product of different genes, which are very different in different individuals, can also contribute to the degree of binding to this cytotoxic T cell. If you just happen to be endowed with a good MHC complex 1 in this type of an infection, then you might get a very robust cytotoxic T cell response. Now, we know that this happens in other types of diseases. For instance, there are other types of MHC1s that are very good in hepatitis B and hepatitis C. So this is well known in other types of viral infections. The question is that the situation here with SARS-CoV-2? And that's a question that still needs to be answered. We're going to answer that here. So let's take a look at a more detailed thing. Here again, we have the target cell here. It's become infected with a virus. It goes to something called a proteasome. This is a little bit more detail, and it gets chopped up into little peptides. Those are maybe 10 to 14 length amino acids. It goes to this tap into this collection area where, notice, right here is the piece of protein fragment from the virus. There's an alpha chain and a beta 2 chain which come together and form the major histocompatibility complex 1 molecule and the peptide together. This goes out to the Golgi apparatus, which then gets secreted out here, and finally makes it right here on the surface of the cell. As you can see here, this cytotoxic T cell comes along with the T cell receptor in green and the CD8, which tells you that this is a CD8 toxic T cell. When that binds, well, there's a signal that goes to the nucleus of the cell that says this is a bad cell, it needs to be destroyed, and there's the release of the granzymes and the perforins. These things latch on here and open up and break up the cell membrane and allow things to come in and come out, and basically the cell gets destroyed. And that's based on this. So the point here that I want to make is it could be the perforin that destroys the cell could be based on the peptides and could also be based on the specific MHC1 molecule. Again, MHC1s are the same in the same cell of each individual, but from individual to individual, they can differ widely. Let's take a look at this study. Again, not peer-reviewed, not published yet, but in the process of it, and it's garnered a lot of attention. This was funded by the National Institutes of Health, and it says here that HLA-B star 1501. So this is the particular gene that makes up the MHC1. So this is the gene, but it can go on to make the MHC1. And of course, you can get a gene allele from your mother and a gene allele from your father. And they're just saying here that this is associated with asymptomatic SARS-CoV-2 infection. So what did they do? Let's take a look here. They have a huge bank, almost 30,000 of people who have signed up for bone marrow transplantation. And so they have already 30,000 people with all of their HLA A, B, and C, and DQ, and all of those for MHC2. And what they did was they asked them to fill out a survey about whether or not they caught SARS-CoV-2, PCR positive, and then what kind of symptoms they had based on a survey. Notice here that out of 29,000, they got about 21,000 people to complete that survey. That's an amazing amount of people that were willing to do that. That really speaks to how dedicated these people are in this study, that they're willing to give blood, knowing that one day they might be asked to donate bone marrow. That's not exactly painless. What they did was 2 phases. They did a discovery phase and a replication phase. This means that they had everybody answer this survey. So the weakness of this trial, of course, is that you're basing it on recollection, answers on a survey. Nevertheless, they got it completed by 15,000 people. Based on these results, what they found was that in the people that were asymptomatic but still positive, there was a huge amount of this HLA B15:01. Then what they did was they applied it to the replication process. In other words, they said, okay, let's replicate this study and look and see whether or not this is the case a priori in the rest of this group. And they found, in fact, that it was very consistent. It was not spurious at all. Let's read the results. I'll quote right from the paper itself. HLA B15:01 was significantly increased in asymptomatic individuals in the discovery cohort compared to symptomatic. And it was pretty large, like 2.5 times more likely to be asymptomatic if you had this particular gene for your HLA-B. And it was pretty statistically significant, and you can see the confidence interval was pretty good. And we reproduced this association in the replication cohort. So in other words, they showed this again in the other half of the cohort with a very similar number, pretty consistent. We found robust association with HLA-B star 15:01 in the combined data set. So when they looked at both of them together, almost a 2.5 increased likelihood if you had this gene to be asymptomatic even though you actually got infected. This is just one allele. So in other words, you get a set of genes from your father, you get a set of genes from your mother, and if either one of your parents gave you HLA-B 15:01, this is what you would have. In the unlikely event that both parents gave you this specific gene, they found here that you were almost 9 times more likely to have an asymptomatic course of COVID-19. They did notice that this other gene here, which has been looked at before, which is a class 2, so MHC class 2, was also associated with it, and it seemed to help if the patient already had HLA-B 15:01. So their conclusion with this study was HLA-B star 15:01 is strongly associated with asymptomatic infection with SARS-CoV-2 and is likely to be involved in the mechanism underlying early viral clearance. I think there has been a lack of understanding out there about exactly what antibodies' roles are in COVID-19 and T cells. We have probably put a bit too much emphasis on antibodies, antibody responses, neutralizing antibodies, and not nearly enough emphasis on T cells. Among participants reporting a positive test for SARS-CoV-2, HLA-B 15:01 is significantly associated with asymptomatic infection. We observed that individuals carrying this relatively common allele, it's about 10% of the population, are more than twice as likely to remain asymptomatic after SARS-CoV-2 infection than those who do not. And an astounding effect for HLA-B 15:01 homozygosity, that means you have 2 copies of this gene, increasing more than 8 times the chance of remaining asymptomatic. This observation suggests important features of early infection with SARS-CoV-2. Let's see why they think this might be the case. They say, although our data did not point to a direct mechanism, compelling evidence indicates that this gene allele is predicted to bind a specific SARS-CoV-2 epitope. And here is this epitope. Remember we said here at the beginning, let's go back and take a look. Remember we said that this protein here is broken up into tiny peptides. Well, one of those peptides is actually very antigenic. And specifically, it's this peptide. And each of these letters stands for an amino acid, but let's just call it the W tag AAAYY. This is a SARS-CoV-2 epitope. In other words, this is a product, this is a little snippet of protein that comes from the SARS-CoV-2 epitope. Actually, as it turns out, it comes from the spike protein. This epitope has been considered a natural candidate for SARS-CoV-2 vaccination studies, as it is considered to be highly immunogenic and predicted to elicit a strong CD8 positive T cell response. A lot of people ask, why is it that we pick the spike protein to use as an epitope for vaccination? Well, this is one of the reasons why, is because this particular epitope not only elicited a good antibody response, but it also elicited a good T cell response. And that's really important, as you'll see here in just a second. The dominant recognition and consequent eliciting of immune response by only a relatively small number of epitopes, that's known as immunodominance. That means even though the virus gets broken up into hundreds, maybe thousands of different types of little peptide fragments, it's only a few of those that's going to cause the real immune response. So they say the dominant recognition and consequent eliciting of immune response by only a relatively small amount of epitopes is determined by multiple factors, including the binding affinity of the peptide for the HLA molecule. Talking about the peptide from the virus with the MHC1, which is coded for by HLA A, B, and C. We speculate that the high affinity for HLA-B1501 for binding our peptide fragment from the spike protein or another unknown viral peptide confers strong CD8 T cell response against SARS-CoV-2 infected cells, leading to better early viral control and thus resulting in asymptomatic course of infection. So that's one of the theories. An alternative explanation is that this particular gene for the major histocompatibility complex 1, previously infected by other common coronaviruses, have pre-existing cross-reactive T cells with high affinity for specific epitope conferred by this HLA molecule. They're saying here that it's possible that another previous coronavirus has activated these T cells in a way that these T cells can then cross over and have immediate reactivity to SARS-CoV-2. Something that a lot of people don't understand is B cells and antibodies will not change. They're very rigid. So when you have different variants of SARS-CoV-2 that infect a population, the antibodies that were good, for instance, for the original strain of SARS-CoV-2 might not be good for Delta or Alpha or Omicron. And that's because of the downfall of B cells. B cells don't have a wide range. They produce a particular antibody. That is not the case with T cells. T cells, as it turns out, will actually change in parallel with the virus in the environment, so that T cell responses are extremely important if you want to keep track of the mutations of the virus itself. And T cells are exquisitely important when it comes to preventing the worst outcomes of COVID-19, for instance, hospitalization and death. Let's go on. While the current literature is mixed regarding cross-reactive CD8 T cells specific to SARS-CoV-2, this might be explained by HLA specificity. The fact that asymptomatic individuals in our study were less likely than symptomatic subjects to report a positive test for antibodies against SARS-CoV-2 suggests a strong early antiviral response and may corroborate the critical role of CD8-positive T cell cytotoxic mediation by HLA class I in eliminating SARS-CoV-2 infected cells. Here's what it means. The T cells did such a good job of eliminating infected cells that the B cells never got a chance to make antibodies against them, potentially. That this combination of this specific gene that makes HLA-B star 15:01 with this peptide that comes from SARS-CoV-2 was such a strong inducer, potentially, of the cytotoxic T cell that it gets eliminated before it has a chance to even get started. That's one possibility. Here's another study that was published back in December of 2021 in Virus Disease. And again, look at our friend here. Here's the W tag AAAYY that was within the N-terminus portion of the spike protein. Cytotoxic T cell epitopes exhibited binding with a maximum number of MH1 alleles. So it seems as though this fragment, perhaps coupled with that HLA-B 15:01, is a very powerful response to T cells. Let's go to February of 2022. This is a paper that was published in Nature Immunology, the T cell immune response against SARS-CoV-2. The adaptive immune response is a major determinant of the clinical outcome after SARS-CoV-2 infection and underpins vaccine efficacy. T cell responses develop early and correlate with protection, but are relatively impaired in severe disease and are associated with intense activation and lymphopenia. That means low white blood cells. A subset of T cells primed against seasonal coronaviruses cross-reacts with SARS-CoV-2 and may contribute to clinical protection. This is exactly what one of the theories was in that paper that we just reviewed. particularly early in life. So early in life, kids, in other words, may be able to use coronaviruses to prime their T cells to effectively fight against SARS-CoV-2 using the same T cells. T cell memory encompasses broad recognition. This is in contradistinction to B cells and antibodies. T cell memory encompasses broad recognition of viral proteins, estimated at around 30 epitopes within each individual, and seems to be well sustained so far. This breadth of recognition can limit the impact of individual viral mutations, like Omicron, and is likely to underpin protection against severe disease from viral variants, including Omicron. Current COVID-19 vaccines elicit robust T-cell responses that likely contribute to remarkable protection against hospitalization or death, and novel or heterologous regimens offer the potential to further enhance cellular responses. T-cell immunity plays a central role in the control of SARS-CoV-2, and its importance may have been relatively underestimated thus far. I think I agree with that statement. T-cell response is really, really important. Let's actually go to the data. And for that, I go here to the Texas Department of State Health Services. We are looking at their internal data in Texas. This is not national data. This is not pharmaceutical data. And we're looking here up to October of 2022. So this is definitely within Omicron. And what they're saying here, that in unvaccinated Texans, they were 28 more times likely to die of COVID-19 associated illness and 13 times more likely to test positive for COVID-19. Notice that this is more of an action of B cells, and this is more of an action of T cells. And that should tell you how important it is, whether it's through natural immunity or vaccination, that T cells are recognized. So let's just take a look at the data here. Here we're looking at people who are 75 years of age, and you can see here these are those that were unvaccinated in terms of death per 100,000, and down here are those that are vaccinated. And you can see here on the scale that we're looking at about 35 per 100,000. Let's move on to the next age group, which is lower, 60 to 74 age group. Here we have the unvaccinated versus the vaccinated. We're up to about 80 per 100,000. Let's go down to the younger group here. We're now looking at 50 to 64, unvaccinated versus vaccinated. Notice, though, that the numbers are coming down. So as we get younger and younger, there's still a difference, but the magnitude of that difference starts to go down. Now we're talking more around like 25 per 100,000. And as we keep going here, the scale is changing. Make note of that. Unvaccinated versus vaccinated, and here we have the 40 to 49 group. Still definitely a difference, but that magnitude is getting less and less. We're up to 16, but still a 16-fold difference here, according to the Texas Department of State Health Services. Here we look at the 30 to 39-year-old group, and we can still see, even in people in their 30s, that there is a significant difference, although now we're down to only 6. And I believe that's all the data they have for deaths, because the amount of deaths in the 18 to 29 and the 12 to 17 are so small, we're not able to even graph it here. This paper used some data that was out there very well. We know already that specific HLA genes can enhance the body's immunity innately against viruses like hepatitis B, hepatitis C. What we didn't know up to this point was whether or not that also happened in SARS-CoV-2. And it looks as though the answer to that is yes. It's interesting to look at this from an immunological standpoint and also from the clinical aspect and critical care medicine. Speaking of critical care medicine, I want to remind you that we have our lectures and videos with continuing medical education, not only for physicians, but nurses and also physician assistants. This is how we get paid. This is how you support us. We're not sponsored by any pharmaceutical corporation or anything else. It's you who support us purchasing and getting the benefit of our medical lectures at medcram.com, and specifically our critical care courses, for instance, EKG interpretation, the Chem 7. We also have a course on vasopressors, the CBC, and how to treat with antibiotics, low sodium, and urinalysis. These are all things that we have on our site, and I encourage you to look at that. So I hope this has been helpful in understanding why is it that some people don't get COVID-19. And I think more research needs to be done on this topic, and a little bit more research probably understanding T cells and why T cells are so important in preventing the worst outcomes of COVID-19. So thanks for joining us.